Okay, this is going to be from Bradford Taylor, emergence of elevated levels of co-infection spatial host microdynamics.
Thanks. Yeah, thanks for sticking around. So I hope to offer some food for thought at least, right? So today we're going to be talking about co-infection and its effect within populations of host microbes. In particular, I'm going to start with a quote from a paper from about 5 years ago. Basically saying 2 or more viruses infecting the same host cell, co-infection, can interact in ways that profoundly affect disease dynamics control. Yet the factors determining co-infection rates are incompletely understood. And so I'm hoping to, help bring some understanding to the factors of that influence co-infection rates, particularly from the influence of population dynamics as opposed to from individual cells. And the reason it's important is these co-infection allows an interaction between viruses. And so some examples of this is direct interference between viruses. So here there's some misfolding in a Mimi virus that results from the infection of essentially a satellite virus. And these particles will now be defective. In addition, prior work has looked at co-infection within a RNA virus and has been able to map it to the game prisoner's dilemma within game theory, when basically stipulating that there's going to be these shared resources and cheaters are able to invade the system. And there's also going to be ecological effects due to changes in life history traits. For example, the birth size, the number of viruses that are being produced during lysis, can be altered as a result of the level of co-infection or the multiplicity of infection. In addition, there's going to be, this can have big effects in terms of evolution, because co-infection actually allows viruses to have sex. And so what happens here is in the host cell, while viruses are being produced, essentially all the constituent parts of viruses are in this pool of resources. So here we've got base plates, genetic material from 2 viruses, and they're in here, and then when it comes time for formation, they basically come together in entropic fashion, and there's a possibility for there to be mixing such that you can have chimeric viruses that are composed of different constituent parts of the different prior viruses. And in addition, there can be direct interaction between the genetic material as well, allowing for recombination. And what this leads to is increased rates of viral evolution as a result of co-infection. So that's basically what happens on the individual scale. So it's important to understand as we build up, how frequent does this happen? And then how do these individual effects affect the population dynamics? And that's what I'm going to be addressing. And so in order to address that, I'm going to take a simulation approach. Now, infection is clearly a local process. You have to be near the person you're infecting. The virus needs to be near the microbe. So I'm going to be looking at an individual-based model on a lattice, where at some time there's going to be hosts and viruses within the community, and there can be an infection event, and lysis is not instantaneous. There's an amount of time that a host is going to be infected before it lysis, and so there can be co-infection at a later time point for the host cell lysis. I'm going to be including, some other important ecological effects, such as host birth and movement, which will be basic diffusion with occlusion for the hosts, so that only one host can be at a lattice point, but any number of viruses can be at a lattice point. Right. And so if we just look at low absorption, first off, we parameterize this considering Prochlorococcus, which is a cyanobacteria in the ocean. And we consider dynamics where absorption is subdiffusive. What ends up happening, I'm not going to show a movie for time, but what ends up happening is there's no characteristic length scale of the spatial pattern. And it's a fairly diffuse system. And when you consider the populations of infected individuals, that is, the hosts with a certain number of viruses inside of them, as the multiplicity of infection increases, the populations match a geometric distribution. And the question is, where does this come from? And so to answer that, we can consider a mean field model. So this is not going to be a spatial model, but one where the system is considered well mixed, and you can come up with simple ODE of population dynamics, which is fairly standard theoretical ecology. And the way that we model it is by considering hosts and then hosts with different multiplicity of infection and viruses. And you can get an immediate result of what the distribution of the equilibrium populations will be, and it's this geometric distribution where there's this scaling probability or scaling factor related to the absorption rate by the virus population and the removal rate of the hosts and the lysis times, or the lysis rates. And so what that means is on this prior plot, this is actually not fit at all. This is just, this is purely from the analysis. So the question is, what happens when we consider scaling up? And so to do that, all right. So to do that, let's first look at the dynamics. And so what you can see is it's very clustered, and there's a spread of viruses, and that these hosts are basically in these communities because the density is going to be, the diffusion is going to be density dependent, and so that they remain in these communities until viruses invade and then basically wipe out them completely. And the question is then, how does this affect the co-infection rates? And what you see is now there's actually going to be an amplification of the densities of increasing multiplicity infected hosts. And so this is the geometric distribution that is predicted from the mean field model, and it is woefully underrepresenting the amount of co-infection. And in fact, even if you fit a geometric distribution to this, what happens is further along in increasing MOI, there's going to be a fatter tail. So what we see is co-infection is amplified, and a higher level of it is as well amplified. And so to get just a quick understanding of that, you can start considering basically the conditional distributions of viral densities. These are conditioned on what host is present in the same lattice site. So what I did was I looked at the host of a certain MOI, and then this is the frequency of viruses of a certain density, and then it's been normalized to 1. And what we see is in the homogeneous case, that was the first case I showed, the distributions are basically all the same. Meanwhile, if we look at the cluster distributions, they start skewing further and further to the right. And the easy way to understand this is the probability of an absorption event to happen in any one cell is going to actually be linearly dependent on the number of viruses present, so that causes this skew. And so ultimately, that's going to be the source for this interaction of the population dynamics and the co-infection levels. And what it leads to is that while the mean field model gives us a MOI distribution that's geometric, what we see is that the patchiness is going to lead us to an increased level of co-infection, and in particular fatter tails, meaning if co-infection has any effect in terms of changing burst sizes, that will then feed back into the system. And the easy way to understand this is that the density dependence in the absorption rate is what's driving this change. And with that, I'd like to thank my lab, our collaborators, and the funding sources. I'd be happy to take any questions. Thanks. Yeah, at this point there's actually no effect that we're incorporating, but we are planning on sort of, once now that we've got the baseline understanding, to start including changes to both the rate of lysis, which has been observed experimentally, and through changes in burst size, which is one of the papers I showed up there.
Okay. No further questions. Let's thank Bradford again.